Hello students, welcome back to the biology class. So in the last class we continued the chapter number 8, that is the human health and diseases, and we covered the topic of AIDS in detail along with the graft as well as the graft rejection concept. We learned that AIDS, which is also expanded in the form of acquired immunodeficiency syndrome, we learned that it is not a congenital disease. It is a genetic problem. Because it is not a genetic problem, that's why we cannot call it as a congenital disease. And this disorder it happens because of the malfunctioning of the cell mediated immunity. The reason behind is the virus which is responsible for this syndrome is called as HIV, or human immunodeficiency virus. This virus first of all attacks the macrophages inside the human body once it gains entry into the human body, and then the macrophages will simply act like the HIV factory, because the virus will first of all make its RNA, that means viral RNA, enter inside the human host cell, and once it enters into the human host cell, it will cause reverse transcription with the help of reverse transcriptase enzyme, and thus the viral DNAs are formed. This DNA will incorporate itself inside the nucleus with the host cell's DNA. So once it becomes a part of host cell's nuclear genetic material, now it will start taking over the whole machinery of the host cell and starts making the RNA with the help of transcription, as well as proteins required for protein coat formation with the help of translation. And because of all these activity, the host cell will merely become a factory to synthesize more number of HIV. Therefore it is called as HIV factory. And once these new progeny viruses come outside, they attack more macrophages, as well as they start entering into the bloodstream, and now they start attacking the helper T cells. So once the helper T cells numbers come down, even because we all have learned that helper T cells are responsible for activating the B cells, so there is a complete breakdown of the immune system. So because it starts with the helper T cell, that's why it is called as that it is a disorder of cell mediated immune system. Now it was first recognized or identified in USA, and then the contribution of Professor Luc Montagnier and Professor Robert Gallo led to the discovery of HIV, and then the name was first of all introduced as HCLV3, which was later changed into HIV in 1986. We learned that it is 90 to 120 nanometer wide, that means the diameter. It is spherical in shape, enveloped virus, because it carries not just the protein coat, it carries the glycoprotein coat around it or envelope around it. So it is an enveloped virus, not a naked virus. And we learned that there are two identical copies of same or similar single-stranded RNA filament along with double copy of reverse transcriptase enzyme. We learned that the glycoprotein coat is synthesized with the help of host cell membrane, and then there are two protein coats inside, and it carries the antigen on the surface, which has receptors on the surfaces of the macrophages as well as the helper T cell. That's why I told in the last class also, cell to cell communication between virus and the host cell takes place. So virus cannot infect, HIV cannot infect all the cells of body. Like for example, it cannot infect RBC because it depends upon cell to cell surface communication that happens only with the help of receptor and the proteins or glycoproteins. We learned that the incubation period for HIV is 6 months to 10 years, so it is variable depending upon the immune system of a person, and on an average 28 months is the incubation period for HIV. Now incubation period is the time duration from the time of contraction of the virus till the point of symptoms to be visible. So it may take a long time for symptoms to get visible, and diagnosis involves ELISA. ELISA stands for enzyme linked immunosorbent assay, followed by Western blotting. So first ELISA is performed, and if the ELISA gives the positive report, then Western blotting is performed so that it can be confirmed whether really the person is HIV positive or not. Treatment involves zidovudine drug, and we learned that treatment can only prolong the death or the life of the person. Death is definitely going to occur before the normal schedule because the person has become a HIV positive person. We even learned about ARC, which stands for the AIDS related complex. It is a mild form of AIDS, and we learned that its symptoms are swollen lymph nodes, fever, sweating at the night time, and weight loss. And patients who have got ARC have higher risk of developing AIDS. So it is something which happens before AIDS symptoms start appearing in a person. After this concept, we started the topic of organ transplant. Yes, we also learned that how transmission of the AIDS takes place and how it does not take place. Then the next concept we learned was the organ transplantation, that is graft. We learned that when a graft is taken from a person and given to another person, there is always chances of rejection. Rejection is possible because the cell-mediated immunity is able to identify between self components and the non-self components. We learned that the first successful graft was made was the kidney transplantation in Boston in 1954, and in India again the kidney transplantation in 1971 in Vellore. We learned that the father of organ transplantation is Alexis Carrel, and there are various types of graft. The first one is when we take a graft of the person and then plant it somewhere else on the body, so there is no chances of rejection in this case. Example, hair transplantation, skin transplantation, this is called as autograft. When the graft is taken from monozygotic twin brother or twin sister, remember identical twins, it is called as isograft. Again, it has great success rate because genetic makeup is same. But if a graft is taken from some other person of same species but a different individual, maybe in the family itself, like elder brother or sister, parents, anywhere, cousins and all, it is called as allograft. So allograft chances of rejection are more because genetically a different person is used. And xenograft has the minimum chances of acceptance or maximum chances of failure because it is taken from a different species. Now whenever a graft is done, to make it more possible to be accepted or minimum chances of rejection, the immunosuppressive agents are given, so like cyclosporin A from Trichoderma polysporum.